being live streamed to YouTube, where it will be permanently archived for public viewing. The audience microphones will be muted for the entire talk, and we also recommend that audience members turn off their camera during the talk. We do encourage you to ask questions, and for that, we will be using the chat feature. Dr. Silvera Siemens, who you will meet in just a moment, will be our co-host and your moderator tonight. And she will be monitoring the chat window for your questions. You can submit your questions in chat at any time during the talk. And then at the end of the talk, Dr. Silvera Siemens will choose some questions from the chat window. Just one moment here. I think I need to sorry, I'm having just a slight technical difficulty. Okay, Brandi should have screen sharing capabilities now. Sorry about that, everyone. Excuse me. All right, there we go. Now we're in business. Okay, so it's about time to get started. Once again, welcome to the Holden Forest and Gardens Scientist Lecture Series. Our mission here at Holden Forest and Gardens is to connect people with the wonder, beauty, and value of trees and plants to inspire action for healthy communities. And we're proud today to be partnering with Black Botanists Week to bring you the next installment of this very exciting year-long series entitled Growing Black Roots, the Black Botanical Legacy. This series will cover a broad range of botanical disciplines and delve into the historical legacy of formally trained and self-taught Black botanists. Through this series, we seek to shine a light on the Black roots within botany, to foster a community of Black botanists, to demonstrate the diversity within this community, and to inspire others who may not have considered botany as a career choice. We want to give a gigantic thank you to the Cleveland Foundation for sponsoring this lecture series. The Cleveland Foundation works to enhance the lives of all residents of Greater Cleveland, now and for generations to come, by working together with donors to build community endowment, address needs through grant making, and provide leadership on key community issues. You can learn more at clevelandfoundation.org. Now, I am super excited to introduce you to my co-host today, Dr. Georgia Silvera Siemens. She's a founding member of the Black Botanists Week Committee, as well as the director of the Washington Square Park Ecopod projects, which conducts monitoring of wild animal and plant populations found within this famous New York City park. And from this platform, offers educational programs and plant biodiversity advocacy. Dr. Silvera Siemens' work speaks to academics and practitioners, which is a topic that is certainly close to our hearts here at Holden. And we are excited to have her here today to tell you a little more about Black Botanists Week and introduce tonight's speaker.
Thank you, Dr. Medeiros, for that introduction. Black Botanist Week is very excited about this partnership with Holden. Black Botanist Week was founded just this past summer to promote, encourage, create a safe space for, and find more Black people and BIPOC who love plants. Our speaker tonight is one of the Black Botanist Week organizers. I am very proud to introduce Ms. Brandi Cannon. Ms. Cannon is a middle school science teacher and curriculum developer, having conducted research focused on the biology of parasitic plants. Her research investigated the host cues that led to germination and successful establishment, as well as the role of disturbances like fire in determining plant rooting patterns and genetic diversity, with an eye toward conserving populations of endangered parasitic plants. She brings her experience and excitement for research directly into her classroom teaching, and we'll get a flavor of that tonight. and is also active in school-based and nonprofit activities that support diverse communities, including leading the Kids of Color Affinity Group and the Middle School WISE, Women in STEM Education Club, Diversity, Equity, and Inclusion Coordinator for the United States Quidditch, and the DEI consultant for the International Quidditch Association. Brandi has organized a list of her top 10 plants by color, aroma, flower type, and leaf shape, and is willing to share with anyone who will listen. So please take her up on her offer. And now I turn the floor to Brandi Cannon.
Thank you, Georgia. Hi, my name is Brandi Cannon, and see here. I guess we'll start here. Identification and discovery, a botanical love story. So I'll start with, I've introduced myself. I'm an 8th grade science teacher. I'm also the middle school curriculum coordinator for science, and I love spending time making science engaging. I like making it memorable, regardless of the experience you've had prior to meeting me, which is really important for me in 8th grade, because 8th grade, it can go either way. It could be like, never again, don't want to take a science class, or it could be like, it wasn't so bad. So I'm tuning in here from New York City. I work in New Jersey, and I hail from the great state of Texas. Houston, Texas, to be very specific, home of Beyonce, Lizzo, and Megan Thee Stallion. But of course, we're going to focus on me, another great of Houston. And this is my story. It's about discovery and love, and spoiler alert, it's not over yet, thankfully. But today, I'm hoping to give you an insight into the life of being a Black botanist who is still growing their own roots in their community. So first thing first, happy Black History Month. I'd like to take root here, or ground ourselves in the knowledge of my people and my ancestors in Black history. Everyone is family, regardless of where you come from, because we don't always know our backgrounds. So we're making and discovering history simultaneously. So I have a question for you, and if you're bold enough, you can try to answer that question in the chat. What plant is this? See if we see any people in the chat. What plant do you think this is? It's okay, you can give me the common name if you don't know the species epithet. What plant do you think this might be? Anybody? Any guesses? Ooh, not quite, but similar leafing. Not a lily, not, oh, the flower just kind of reminded me of a daffodil. Yeah. Ooh, we've got some dead on. Yeah, it's definitely a type of orchid. It is a vanilla orchid. Absolutely. So moving on to this, I want to focus on what's in a name. And I really thank you all for joining me, and I am going to try to be as engaging as possible, because that's partly what I do as a living. So what's in a name? We'll start with Carolus Linnaeus, who is the father of names, first of his name, mostly because he rendered his own name to be actually Carolus Linnaeus. His name is actually Carl. So Carl came up with this really great system. And it's organized in a way that allows us to classify organisms, and we call that taxonomy. So we use this really cool thing called binomial nomenclature. It puts a genus and a species together, so it allows us to specify and organize things by common traits, behaviors, and it kind of determines where you come from and where you're going sometimes. So a genus, if you're not familiar, it organizes organisms by common traits, and the species part of it sort of describes a more specific way to organize by common traits. So a lot of you guessed correctly that this was Vanilla planifolia. Vanilla being the genus, and its meaning comes from vena, which is sheath in Spanish. And planifolia, if we break that down, plan is flat, and folia means leaf. So it's a flat leaf, which you could kind of see at that first photo. And Vanilla planifolia comes from the Orchidaceae family. So This is a family that's one of the largest ones in the world, right? It is rivaled by the Asteraceae family, what we know as like the sunflower family. Yeah, you're probably right. Almost everything feels like it's a sunflower or related to it. So by grouping these things and organizing them, we can sort of figure out where things go. And also, like when we're going on botany trips, it's a little easier for us to figure out what things are. So let's do a little bit of a practice. Personally, I find most folks have a familiarity with animals. I'm more of a plant person, so this will be mostly a plant talk, but I figure we'd give it a try. Gorilla, genus and species. It's not too out there, pretty simple, right on the nose. I recently found out that in 1799, the Naval Cron wrote that the actual epithet of gorilla, where it came from, and I was kind of taken aback by it, as I am when I'm researching most things. It means tribe of hairy woman, and that really comes from these colonizers, these European explorers who went to West Africa and wrote down these accounts of coming in encounter with these savage women who were in these tribes who were very hairy. And I'll let you do your own research on that one, but not too friendly. I definitely say what that encounter was like. And unfortunately, that happens to be what a lot of being Black and doing research and exploring into science is about. So gorilla, who knew? Spongiforma squarepantsii. This one's a fungus. It's a sponge-like fruiting body. It has a sponge-like fruiting body. And apparently when you squeeze it, water comes out. So go figure. But not too out there, the animal folks and the fungi peeps. But I think it's a pretty funny name. It doesn't really tell us what it really is, though. It's not like flat leaf. My favorite, Felis catus. This one exists in my own home. Her name's Vanta Black, named after the darkest substance known in the world. This one, cats are strange creatures, but Felis catus just kind of gives it away, right? It must be a cat. And lastly, and this one, I'm going to ask you to really think about it. These two. When we're thinking about what's in a name, you probably already have a name for this. So I'm going to ask you a question, and if you're here joining us in the Zoom, I'm going to ask you to go ahead and give it a shot on this poll. Which animal do you think is a Black Panther? 1, 2, and for kicks and giggles, we'll throw in number 3. Which animal do you think is a Black Panther? Number 1, number 2, or number 3? Number 3 thinks she's a Black Panther, but she's not. We had a tie at first, but now it's like bouncing back and forth. They are not the same animal. I will give you that. Interesting. We're like 40-60% right now. Still 40-60. Okay, so let's stop there. So 39% of you think that number 1 is a Black Panther, and 61% of you think that number 2 is a Black Panther. Not too bad. Nobody thinks number 3 is a Black Panther, which is okay. Kind of what I was expecting. I want to let you know that neither of these are Black Panthers. And, it's all about what's in a name. What have we learned to sort of name Black Panthers? Well, it's a black and it's probably a really big cat. But neither of these are Black Panthers. Of course, the genus name is Panthera, but Panthera just recognizes large cats with very specific cranial features. But these are actually 2 different types of cats. Number 1 is a Panthera onca, which is actually a jaguar. It's a color variant that it experiences where it has excess Black pigments. So you can only find this one in Americas. So North America, South America. This one is actually at a zoo. He's got a big body. Number 2 is Panthera pardus, and Panthera pardus is actually a leopard. So that leopard is, again, it has that excess black pigments. And it's actually really interesting because number 1 is experiencing a dominant trait of black, and number 2 is experiencing a recessive trait of black. So, but you still get these like Black Panther sort of traits here. So there is technically no such thing as an actual Black Panther. It's just that these cats got their names because of what we sort of categorize them in layman's terms, or in normal people passing. So why do we really care about this, or why even bring this up? Or as my 8th graders might ask, will this be on the test? And I think it's part about misidentification. Naming things is tough. Categorizing them can not always fit the mold of what we're looking for. And part of the story is about truly understanding that organisms and people, as they are before boxing them into these sort of labels and tucking them away. And I think some taxonomists might actually agree with that. So going back to the vanilla, also known as the black flower, I believe the pronunciation, and I will try my hardest to get this right, it's Tllil Xochitl. It is known as that this black flower in pre-Columbian Mesoamerican people who first cultivated it, the vanilla orchid. Once it matures, though, it creates this nice shriveled up fruit, and it turns black, and that's what we know as the vanilla pod. European colonizers attempted to cultivate vanilla outside of Mexico, and they failed at every turn, basically trying to capitalize on its production, because this was definitely knocking custard out of the park once you put it in there. Unless this is pollinated by its local bees, the flowers can't pollinate themselves. And that's where our story gets really interesting. So, like I said, these colonizers, they took the opportunity and took from this area of where Mexico currently is. And that takes us to La Réunion, which is a French republic east of Madagascar. So that's where the "you are here" red little dot there is. It's said to have a diverse population because of imported workers brought from all over the world. Let's be real, that's definitely enslaved people. And when we're talking about this, like, it was kind of mind-blowing thinking about this, because you're not really taught a lot about African history, right? Or it's sort of reduced to like Egypt, or it's reduced to maybe some like folklore or mythology, but you don't really get like deep into this sort of what's going on all around Africa. So as I was reading this, through my research, I found it had a diverse population afterwards, right? There are Chinese, Malagasy, African, European, Indian folks all working there. It's just the non-Europeans were the people who were enslaved. And this island is very small, right? 39 miles wide, on top of a hot spot, beaches galore. And yet you meet this interesting character there. His name is Edmund Albius. In 1841, this orphaned, enslaved 12-year-old boy figured out how to cultivate vanilla, right? So they've taken it from Mexico, they bring it over to this French island, or French Republic, and he learns how to make vanilla bean pods, which people were like, what? How did you do that? And after the abolishment of slavery, he goes on to be a kitchen servant, and he eventually dies in poverty. And this just like blows my mind at the time that I'm finding this out, because vanilla is a $65 million industry. And if $65 million doesn't seem like a lot, right? Maybe it's not what people made on the GME stocks last week, but $65 million for a plant fruit. Can you imagine? We would be messed up if they did that to bananas. So, I'm thinking about this, and I'm learning about what Edmund's going through, how he learns how to take a blade of grass, and he realizes that there's this membrane in between the male and the female part of the flower. And he moves that, and he squishes it, and he smears the pollen together, and then voila, you get a fruit. So, this young boy is doing this, and he gets no credit. It's not too surprising though, right? It's 1841. But what's interesting is the motto for this place. And this is the thing that really like it carries me as I'm like learning the story. The motto is Florebo quocumque ferrar. If you know French, maybe you give you a second to try to figure what that means. So I will flourish wherever I am brought, which is really cringy in the fact that slaves are here, but it's also kind of inspiring for realizing what a lot of enslaved people went through. I will flourish wherever I am brought. So regardless of what's going on in this colony, right? Somehow this is Black history in a nutshell. It's everything I've been taught by my elders. It is, I will be amazing. I will find amazing things. I will do amazing things wherever I am. And that just really stuck with me, especially because I learned about Edmund Albius in 2020. That's right. When we were all stuck inside. Oh, wait, we're still stuck inside. While I was stuck inside, I was 30 years old. I'm like, how did I not know about this Black botanist until this year, right? And then it makes me think about, you know, what do I not know? I only found him because I was digging up potential social media posts, social media information for my Twitter during Black Botanist Week. And I was like, oh yeah, let's find out the roots of vanilla. That kind of took me here. So that stuck with me for a bit. You will flourish. So what's in my name? Well, first there's Maddie Williams and Papa Cannon. He goes by Cha. Both from Arkansas. Maddie Williams, a brilliant social worker, an educator, and Papa was a veteran, a bank messenger and a bus driver. I only saw the bus driver tail end because that's when I was born, and I loved getting to sit on his lap and ride in the bus. That was like totally my thing. And what's really interesting about this story is that they didn't meet in Arkansas. They met in East St. Louis. The story continues with Jerry Edwards, who is an avid gardener, a huntress. I truly mean that. She hunts, and a local pastor. And Emmett Edwards, who was a generous and kind custodian from New Orleans. Jerry Edwards was from Alabama. Emmett was from Louisiana. And somehow they also met in St. Louis. My grandmother is Jerry Edwards. She's the only remaining grandparent alive. And she likes to tell me about this picture because she used to sing in jazz clubs in New York City. So she likes to like chuckle about that and say how she used to show her ankle. But, you know, it's really interesting looking back because for the longest part of my life, this is all of my relatives that I knew. And not because I didn't want to, but because lines in our Black history had been broken. So all hard workers, and they all definitely worked with cotton, and that really hits me, right? Like they've all somehow, some way, either had to work with it before school, had to quit school to just work with it, and then they lead us to these two, the high school sweethearts. Those are my parents, and all of them gave rise to me. And I think that's really important when acknowledging Black history and what it means. I am the descendants of laborers and community builders. And I see that in myself now. I didn't really see it before, but as I'm starting to like put puzzles together as I was making this presentation, it also really started to like bubble down so I knew who I was and what I was becoming. So now you know how I got here. And I'm not going to have to render my name in Latin for you to get that. It's a special story about a Black flower, if you will. So what's in a name? Brandi Shantia Cannon. Brandi, my mom wanted a unique Black name in 1989. Shantia I was named after Shanti or Chantilia, which was my mom's favorite perfume when she was pregnant. She no longer likes it, but that is neither here nor there. And then Cannon, the family name. But for the longest time, you could not convince me otherwise that my name was not synonymous with being Black ain't easy. Like accurate, right? That's all I knew. So it's really interesting that, you know, when we go to this, I think about like my childhood. I always loved science, always curious. I would sit and go through all these National Geographic magazines. I would, just sort of immerse myself in things that I couldn't do. I always asked to go to summer camp, the science summer camp, and I couldn't because my parents couldn't afford it. And there was just no way that, the kids had to be watched. My parents had to work. So look, that was what it was. But I found science other ways. My mom is a huge gardener. So my grandmother, she's a gardener for nourishment, I would say, being able to refrigerate and pickle things and she actually hasn't been out like since COVID started because she's so set with like all of her jarred stuff. But my mom, she likes to call her garden the garden of vanity. And it's just all these beautiful flowers. And here are actually some of the flowers. So one of the things I really liked about this is that, looking back, I realized my mom was a scientist. She didn't finish school, but she was a scientist in her own garden. She had journals. And when I say journals, I mean like 2005, 2008, 2007, everything. And she labeled, what the humidity was like every day and when the flowers bloomed and when it was time to rotate the flowers for the seasons. And I didn't really realize that this was a science until I went to college and I started taking botany. So, she eventually went on to make her own landscaping business and took what she knew and was able to put it into something. But I think it was really interesting for her to do these things that she had maybe learned from her mother in part, but not even realizing that there was some science, some history within her own garden. I say that because the picture in the bottom left is Caesalpinia pulcherrima. I don't even think she knows this. I didn't know this. It's known as the peacock flower or the pride of Barbados. The seeds of this genus are poisonous and were used by enslaved Africans as abortifacients, right? So this is this beautiful semi-green, it can get up to like 5 meters tall, gorgeous flowers, yet it's holding the seeds for something really deadly, something kind of scary, actually. So I was doing some research. I found in 1705, this German botanical illustrator, Maria Sibylla Marian, she notes this. She notes that this plant was an act of resistance. It was a reclamation of their bodies and reproductive processes. Enslaved people were using this to remove their future children from the fact that they didn't want them to be enslaved. They were reclaiming their bodies because they were being forced against their will to produce more potential for a labor force, for masters, for their owners. Botany just became something way bigger than I could possibly imagine, right? I knew about the medicinal treatments. I knew about using herbs, right? But this was in my own garden. And that felt different. So I'm thinking about this act of resistance, reclamation of their bodies. Well, I won't lie. Unsurprisingly, I was also in active resistance as a child. And this is why. I hated weeding. I absolutely hated weeding. And my mom, she used to be like, just please help me weed my garden. And this was the yucca bed, the infamous yucca bed that I sometimes write about, because there was sago palms in there. There were tall yuccas, small yuccas, little cacti that could poke you. I was just like, I hate this. Because in Texas, in the middle of the summer when the weeds are spry high, the last thing you want to do is be on your knees in like 100-degree weather trying to figure out and pull out all the weeds. So it's really interesting that my background actually comes from not liking plants. I was terrified of bees. I wanted nothing to do with them. Weeding the yucca bed sucked, and me and plants just didn't mix. You can see me here. I look so sad. So that's how I knew what I did and didn't like. But how I saw myself was really complex. I referred to myself in a college essay as a mahogany shrub, wide and dark, stiff and reduced. I I've been thick with two C's for as long as I remembered, right? Avoiding lecherous gazes from guys who were much too old for me. I'd been categorized, identified, boxed into labels that left me in a daze. Am I a girl? I don't like girl things, right? Am I Black? But my skin is the color of espresso beans. I wasn't even really into rap. So am I Black? Right? These are things that were going through my mind a lot as I was growing up. And I think the worst part about it was all that I knew was that suffering was to be had because I was a girl and I was Black, and those people suffer the most, and I hated it. I will never forget a moment my dad said, "You're Black and a woman and intelligent, so you're gonna have to put in twice as much effort and make your success twice as good as others just to be recognized for how great you are." That is something I continue to carry. But I did it. So I went to Sam Houston State University in Huntsville, Texas. My first science class was botany, and I actually really liked it. I think my prior experience working with plants both in and out of the garden, and now reading about them, I was able to tie those things together in a way that I wasn't expecting. I reached out to Chris Randall, who I was like, look, I don't even know what I can do. Can I do extra like work, or, how can I get involved? And, at the time I didn't know that research labs existed. I just wanted to learn more. So he took me into his lab and said, look, we've got some herbarium samples. We need to like organize some stuff. If you want to do it, you can. And I did. And that's when I fell in love with parasites. So I worked with leafy mistletoe under Chris Randall in his lab for quite some time. I studied Phorandendron leucarpum, which is this lovely plant here. It is an aerial obligate hemiparasite. Let me break that down for you. Aerial in the sky, obligate meaning that it has to parasitize something so it can complete its life cycle, and a hemiparasite meaning that it still has chlorophyll inside of it, right? So it can still photosynthesize, still bring in nutrients, but it has to parasitize to get the rest of its nutrients. So an aerial obligate hemiparasite that invades woody plants to obtain water and minerals. Seeds are dispersed by birds who excrete them onto trees, and then seedlings become fixed onto branches, and then they form a root or a specialized root where they parasitize its host. So let me tell you, I knew about mistletoe because people kiss under it, right? But I didn't really realize where mistletoe was until someone opened my eyes. This is mistletoe. Have you ever gone by, and right now is like perfect mistletoe season. Have you ever gone by a tree and seen like these big tufts of green? Sometimes you'll see like one or two. You're like, there's no way that can be a nest. And then you go about your day because you don't really care. This is mistletoe. And this is exactly what it looks like. And now that you have the X-ray vision, you can never take it back. Like I said earlier, mistletoe it basically gets stuck to birds. Right, it's got this really sticky aril coating, kind of like the aril or that red gooey stuff on pomegranate seeds. Birds ingest it, it gets stuck to their butts, and then they have to wipe their butts on trees to try to get it off. So that's the beginning of a mistletoe seedling's life. And I think it's really important here because there's an importance in all life, right? All diversity. But even with parasitic plants, and I think that's the thing that really got me. This Cassilia indivisa up here at the top right, that is a parasitic plant. Most people wouldn't know. Gorgeous, pink, stands out, takes over everything in Texas. And yet somehow you're like, but that's not what I've categorized to be a parasite. So parasites develop this haustorium, right? It penetrates vascular tissue of the host plants. It can parasitize either the root or the stem, and it uses it to obtain nutrients and water. Not all parasites are horrible, right? Sometimes they just need support from another plant in terms of getting water or nutrients, and sometimes they can absolutely devastate crops. So I don't want to give you the sense that it's all great. Cuscuta pentagona is one of the parasites that we used to start our research. We didn't work on it, but we actually read papers about it. And we started thinking, well, if Cuscuta is like this kind of like wily plant that grows and parasitizes things, but it doesn't do it by air. It just does it by like stem and root, like it's on the ground mostly. But how do aerial parasites find their hosts? And that's when things get weird. So I'm going to tell you a little bit about that research, and yeah, I just hope you enjoy it. A key here is that it just, so you know, this is the parasite hostorium that's coming in, right? And this is actually the host root. So you can actually see the tissue sort of like parasitizing there. It's creating the xylem bridge where it can sort of tap in for nutrients. This is a lovely slide by Jeff Morowitz who got that for me. All right, so my hypotheses for my research were to establish contact with a host, plant hostorium must sense and respond to host signals. So if that is true, right, or potential ways to support this is by investigating whether emerging seedlings detect hosts by light cues, right? We know seedlings don't have eyes, but can they detect light? Hypothesis 2, emerging seedlings detect host cues or host by chemical cues associated with a physiochemical substrate, right? So does it matter if they're on the host or not? And then number 3 is emerging seedlings detect hosts by airborne chemical cues. So this is the idea that maybe they're sniffing their hosts. So we started here, and this is where we worked on St. Houston's campus. Most people just take everything on a tree hanging off as an art project, so that wasn't too bad of a problem. But what we did was we collected fruits, we germinated them in hydrogen peroxide, we let them dry, and then we began our experiments. So hypothesis 1 was testing for light cues. So, a trip to Walmart, got some tulle, lightly put it over these seeds that we glued to trees with eco glue. And the treatment was actually covering it with a dark cloth. Then we had our second hypothesis was, does it matter if it's on an actual branch, whether it needs to penetrate a branch, or can it be on, say, cutouts from a plastic bottle? And, will it try to form a hostoria there? Hypothesis 3 was about smell, right? Is it smelling its host? So what we did is we planted these little lovely seedlings on dead branches and then spaced them between living branches. So the living branches right here is up top and the dead branches on the bottom. And there you can actually see this is that little radical growing towards, and the radical is just the root. It's actually growing towards the living host, which is probably the most adorable and most like scientifically satisfactory thing that's ever happened for me. So what we did here is also, just in case you wanted to know, we did plant all of these on Quercus nigra, which isn't black oak but water oak, but neither here nor there. So our data here shows us that establishment, so we have viability and we have establishment. Viability meaning how long did it survive, and then establishment, did it actually make contact and sort of form a hostorium. So the seedlings died without light by the 15th week, but we did notice that results supported that seedlings sensed light, right? Because it seems like when you removed light, the establishment dropped off. When it comes to substrate, viability, there was no significance, right? It didn't really matter whether it was on the plastic or on the tree, but the establishment, when it was placed directly on the bark, there was significant establishment. And within the second week, you can actually see that there was continuous support, that seedlings detected some sort of chemical cues associated with the substrate or the bark. And then with the proximity, viability plant shows that plants uniformly grew towards their host, right? They even though they couldn't make, he can't make right here, couldn't make that 5 to 10 millimeter gap. There was no significant difference in the establishment though. So after this happened, Chris asked me to head an experiment by myself. At this point, I'm like, what? There's no way. You've got the wrong person. I don't know what you're talking about. But he said a lot of nice things that people do when they trust you. And I assume he trusts me not to mess up. So I pushed this forward for my senior thesis. I studied, I took the mistletoe, and I wanted to know whether the hostorial growth, right? So that's a specialized root that penetrates the plant growth, is influenced by volatile chemical cues emitted by its typical host, right? So this is where things get weird, right? I'm basically trying to see if these seedlings can smell. And that is probably one of the weirdest and coolest things ever. So I'm reading papers, I'm reaching out to PIs, I'm looking for advice, and I'm coming up with plans. I'm doing science. And we look at these monoterpenes. So monoterpenes are listed here: pinene, limonene, myrcene, and phellandrine. They're basically these volatile compounds that trees and plants produce. Sometimes you actually use them in your essential oils, right? They're the organic byproducts of metabolism. And it's interesting because we read that they were inducing growth in this other parasite, Cuscuta. So my lab mates and I constructed this thing that you see here, this lovely PVC shelf with wooden rods. It held about 60 Petri dishes. And our Petri dishes were actually where we put our seed on these lined filters. You can see here that we actually put in these different color pipe cleaners, and the pipe cleaners represented different types of monoterpenes. So you either had a blue and pink, a blue and yellow, a blue and green, or a blue and purple, blue being the control, right? And then we had an entire control group. So we kept them moist, we watered them, and we just let them grow. We would replace that monoterpene every day because it did evaporate, right? Because it was diluted in a sort of alcohol. But what was interesting is that, as you see in this bottom right corner, you can actually see it growing towards a certain way, which is like kind of nuts, right? So we checked this, we looked at the pictures randomly, we searched for viable seedlings, took pictures, double blinded this test, and we basically took the radical length of the primary root, which was recorded from separating at a right angle, right? And you can see if it's negative, that means it grew towards the control. So this one was kind of like that. And if it grew towards the right, or if it was positive, sorry, that meant it actually grew towards the monoterpene. And this one was weird. It just went straight for the paper. So with this, we did significant test treatments, and we did with an ANOVA measuring distance from control. You can see that the far right three, there was definitely preference, right? We're seeing preference and smell basically, as opposed to just the control and pinene, which if we go back, this is pinene and it grew very much away from it. Like that is massive. It's massive growth to be like, I'm going to put the energy in to grow away from the smell. So yeah, that was pretty huge for me. I'll forever remember Chris Randall's lab as a place of growth for me, finding my own 2 feet, and just being. I think that is a great way to hold a great story to hold in my heart, because they definitely don't tell you when you leave school just how much imposter syndrome hits. So does anyone know, I'm going to put this last poll up. Does anyone know what this Pokemon is? Who's that Pokemon? Right off the bat. Yeah, y'all know it. So, I didn't go straight into grad school. I think my mental health was not in the place where I should have gone. You could say it was trubbish, if you will. I left school feeling like rafflesia, but I felt reduced, right? As soon as I left school, my worth was limited to, again, my gender and my skin color. I felt like Vileplume, and Vileplume is the correct answer to that. It wasn't a good feeling. I want to go back to vanilla really quick, and I'm not going to read all of this, but I just want you to get a sense of how difficult it is to nurture vanilla. 50% shade, 80% humidity, got to grow between 10 and 20 degrees latitude, moderate rainfall, evenly distributed between 10 months. And of course, it's an orchid, right? So it has to grow between very specific temperatures. It can grow as tall as it wants, but it may not produce flowers. It felt like me at a time. Cultivators have to fold the vine over to promote more flowering, which felt like my lab advisor at the time. Self-pollination is blocked, right? I told you a little bit about how Edmund Albius had to take that away. Vanilla flowers only open for about 12 to 15 hours. Insanity, right? 12 to 15 hours, then you lose your chance. Fruit takes 5 to 6 weeks to develop and then 6 months to mature. Starting to realize why vanilla is worth so much. And vanilla seeds won't even germinate by themselves. Most of these are just cuttings, right? Because vanilla seeds actually need a presence of a mycorrhizal fungi, right? Because basically orchid seeds are so small that they have no internal nutrition by themselves. So they use these mycorrhizal fungi to help them get enough energy just to kickstart off. Talk about having to nurture. This is almost babying at this point. So I weathered through, right? I weathered in the outside world. And that's not to say that I didn't do really great things. I got the NSF REU internship to Cornell. I got to go out of the country to do sampling and plant stuff, which was really great. I won awards for presentations, but for some reason, it still felt like I couldn't own my successes. I was chasing after my dreams. And maybe that was because I couldn't see myself. So I took to gardening. And that's probably the weirdest like transition of anything, but it really grounded me in a way that I wasn't expecting. My mom gave me a patch in her backyard, and my family had squash for the first time, grown by these hands. So I wanna hop, skip, jump a sec, just so I can get to the point that I want to. Here we go. So I'm gonna jump really quick to something that I thought was really inspiring and something that really motivated me. So Georgia, I'm gonna have to ask for your help if you don't mind.
Okay.
All right, Georgia.
My favorite TV show. I'm ready.
All right. Can you pick a number on the board? Pick a topic and a number.
Oh my goodness. These all look really good. Okay, Botany 101 for 300, please.
Okay. All right. So this Black botanist was the first woman to earn a PhD at Fordham University, and went on to head the botany department at Howard University for 29 years. Anybody know? You can try it by putting it in the chat. Anybody got it?
Looks someone right off the bat posted an answer.
Yeah, that's amazing. Right off the bat. Yeah. Great job, Catherine. All right. There we go. So Dr. Marie Clark Taylor, who, again, I didn't learn about until my 30s, but you know, there's a lot of growth for learning. I'm all here for it. Was an academic, earning her BS and MS at Howard and then a PhD at Fordham. First woman in science to earn a PhD, which is wow. She's a researcher who focused in plant photomorphogenesis. So that's how plant growth is affected by light, not necessarily in like which way it grows, but how light can trigger certain parts of plant growth. So whether it's like flowering or leaf growth and things like that. She started a summer science institute to teach science instructors how to use live plant material in microscopy when teaching students. She served in the Red Cross during World War II and was personally invited and requested by President Lyndon B. Johnson to expand her work overseas. When I found out about her, I was like, whoa, that's who I want to follow. And I was incredibly inspiring for me because, part of being a Black botanist is making your own path, mostly because you don't know who came before you. And I think finding out about Dr. Marie has sort of reinvigorated my own passion, right? I love teaching, and I love communicating science in a way that kids really enjoy. I love taking away these things and telling people about them, because where else would we be if we didn't know about it? So thank you, Catherine, for getting that one. I could go on for days about the type of research that I've done, and the type of work that I continue to do. But I want to end here with what will be your legacy. And I take a lot of this and I think about, what's inspired me. Has it been great teachers? Is it my parents? Is it my elders and my ancestors? Is it my kids I teach every day, right? That stuff really keeps me going. And I think a great way to sort of end this is to remember, I found this and I thought it was really impressive. It's the myth of the Negro past. And it's quoted, millions of Africans who were dragged to the New World were not blank slates upon which European civilizations would write at will. They were peoples with complex social, political, and religious systems of their own. By forced transportation and incessant violence, slavery was able to interdict the transfer of those systems as systems. None could be carried intact across the sea, but it could not crush the intellects, habits of mind, and spirits of its victims. They survived in spite of everything. Their children survived, and in them survived Africa. So this is currently my love story. It's about growing, developing, evolving, right? It's about learning to know myself and my being, right? I describe what's Black. I describe what my gender is. I become who I want to be just by being, and full of self-discovery, right? Uncovering my roots I didn't know were intertwined with my own culture and heritage. I'm growing with plants, I'm tending to my soil, I'm giving myself light and nutrients. It's planting seeds in a garden you'll never get to see. But as long as I'm planting seeds, I know that they'll eventually be there, right? So I want to thank you so much to Holden for allowing you to share my story. And I want to thank you to my friends and my family who've continued helping me grow. And I also want to dedicate this presentation to my friend David, who passed away from COVID, but had a huge amount of love and passion for growth in plants and sharing his love with cacti. Thank you.
Wow, Miss Cannon, I'm feeling a little choked up here. You just ended on a really deep note. That was wonderful. I can see your years and years of teaching, because that was just a glorious presentation. Thank you so much. And I'm going to try not to just go on here myself, because I know Dr. Silvera Siemens has some questions for you from the chat, and everybody's dying to hear more from you. So I'm going to turn it back over to Dr. Silvera Siemens.
Yes, there are some questions, and I just wanted to say my own personal thank you to Brandi for sharing this powerful portrayal of Black botany. It's really wonderful to know that a Black person was involved in the cultivation of the vanilla bean, which is this treasured spice and quite an expensive one to boot. So that's definitely something I'll never forget, and I'll certainly pass it on to my own kids. Thanks for sharing your family tree, that your mom is part of this legacy of like amateur scientists and phenologists, but we're not really amateur, have like fantastic local knowledge. Your botanical journey has been thorny, it's been exciting, and it's been inspiring. And I'm so glad that you shared all of it, or up until this point, with us. So I'm gonna jump right into the questions. And this one is from Sue. Your passion for your research is infectious. Do you have any plans to let your students participate in your research?
Absolutely. Maybe not this research specifically, right? Because it's based in Texas. But that is the next step, is getting middle school and high school students to do research. And not just like, oh, does the plant grow towards the light? I think that's great too. But I'm really interested in this idea that you don't have to wait to get to grad school to make a difference and do this sort of laboratory and research science, especially since a lot of the science I did was based in things that you could just buy from Walmart. It's all about having the right knowledge and being creative enough to use the right tools.
Okay, we have several more questions. This one from Sarah. Do scientists know if the volatile compounds you tested are key for the mistletoe to parasitize its host? For example, do increased terpenoids deter the parasite?
That's a great question. And I wish I could answer it like with certainty, because that was the next step that the lab was getting to, but I graduated. So I can't tell you for certain if terpenes are definitely turnoffs, but from what we learned in our experiment and what we've read in papers, chemical volatile cues are definitely involved in sort of directing growth of the radicle and then the hostorium, because you wouldn't see any of those little parasite seedlings mounting themselves on anything that was dead and wasn't emitting anything. So absolutely, I think that definitely has something to do with it. I'm not sure if I haven't read like the latest research, but I'm not sure if they've what's the word I want to use? Narrowed it down to specifically what is that thing that's getting them.
Thank you. I just wanted to let you know there are just so many compliments about your presentation that we could probably spend the next set of this presentation reading them off. Another question, and it's related to the one about your students, so I'll sort of jump down to that one. What are the biggest barriers to including more K-12 students in research?
Yeah, I would say the biggest barriers for K-12 research is not trusting that students are capable of challenging themselves. I find that the biggest barrier I face as a teacher is when I'm working with some educators, that they're worried that students are too overwhelmed or that they don't have the tools, when in reality kids are probably the most flexible beings in the world right now. Especially you see that a lot with like working with kids in COVID, right? It's like every other day we're either in school, out school, hybrid. They're just so flexible, and I think their curiosity and, let's be honest, the amount of free time they have lends a lot of moments for them to grow and challenge themselves and really push themselves forward. I think a lot of kids don't remember what challenge is like because they are not challenged. And once they see that they've conquered a challenge, they're like, I can do anything. So I think it's just motivating students that they can, like, the sky's the limit for them, but also, being an assistant and a guidance and a tool for them when they need it.
Thank you. It sort of means a lot coming from someone who's like steeped in this work as an educator, to sort of hear your perspective on students' abilities. Our next question is from Mary. Can you talk a bit more about finding motivation to do science when you're feeling, and this word is in quotes, diminished?
It's rough. I found, especially in grad school, I found solace in my friends, the people in my cohort, the people who were going through the same thing I was going through. But I also find a lot of, I think, help, like just talking to my mom. Like I talk to my mom a lot. I'm not gonna lie. Sometimes my partner's like, you're still on the phone. It's been like 2 hours. But like talking to her brings me some sort of like, it makes me feel calm again. And if I feel calm, I can feel like I can try to approach things again. And I think always remembering, like, you know, that imposter syndrome gets the best of us, but like reflecting back and thinking about all the things that you've overcome, you're like, I can absolutely do this. It's going to suck and it's going to hurt and I might cry, but I'm going to do this. And I think that's the thing that really gets me over the part about feeling diminished.
Thank you. I hope I won't let my mom know that I have a friend who talks to their mom for 2 hours at a time. Another question, this one from Juliana. You mentioned that you're still growing. Can you talk about how you see your work on the Black Botanist Week committee, how that will influence your journey going forward?
Yeah, I mean, wow, Black Botanist Week. You should have been like the first shout out at the very beginning of the slideshow, because I don't know how many Black botanists I knew by name before last year. And now I just have like a plethora of you. I can reach out to you if I have questions or I'm struggling. Or Georgia, we had a social distance botany hike in New York City. Like, what a moment. and so many shared experiences, but yet so many things that we can learn from each other. So I would definitely say that has been the biggest thing for me. And then finding other Black botanists, right? That's the goal, and sort of bring a spotlight, just like the Black at all the other science social medias platforms that have been put out. But finding people who are going through similar situations and people that you feel like you can trust, right? Because like I said in the presentation, right, we're all family. Even though we may be coming from different backgrounds, we have a lot of shared experiences that put us in a place that is unusual and unlike, I think, many other situations.
I have a question for you, Brandi. So you've discovered that plants smell, which I think is fantastic. And I'm thinking about the sort of recent focus on the ways in which plants communicate with one another through their fungal networks, and the possibility that plants are communicating to, non-plant organisms. And I'm curious how you situate your findings that plants smell in the sort of philosophy or line of research about plants as more than sort of sessile, immobile organisms. And then a second to that is, what would you like to discover next about plants?
It's a great question. Questions. Plants not being sessile is like, that's one of the things I love telling kids about, because they're like, plants, it's not a sloth or a unicorn. I'm like, unicorns aren't real. But plants are amazing in their own ways. A lot of my favorite plants are the ones that move, like the Venus flytrap, or when you think about the largest organism in the world being poplar trees, no one would ever think that they're alive and growing and moving, right? And I think that's one of the things that, like, when I was like, plants smell, like, this just gives me more of a reason to sort of immerse myself in this idea that plants are so much more, so much bigger, and they'll be here after we're long gone. Their importance and significance in our relationship with them, especially when you talk about like this community of plants and how they communicate with each other and the things around them, right? That's all ecology. And it may not be the exciting ecology of the the rare panda or the animals that we're trying to save kind of thing, but I think it lends us a reason to say, hey, this is just as important, this is just as diverse, this is just as significant. So I would definitely say that. And then in terms of what I would like to research next, who knows? My grandmother is always telling me to find a cure for arthritis. I'm like, Grandma. But I think I don't really care about what I discover. I think I want to be a part of the process. I just want to be there and just be just as wowed as I was the first day I realized those tufts trees were mistletoe.
Thank you so much, Brandi.
Thank you for that great question to end the question and answer period. Ms. Cannon, I am just going to reflect what someone else said. I wish you were my 8th grade science teacher. because this was really fun. I had a great time. Unfortunately, I think we're kind of out of time. I want to remind everybody that you can watch this talk again and again on YouTube, and we've developed some educational materials that go along with the lecture, which you can find on the Holden Forest and Gardens webpage. Dr. Silvera Siemens has put that link in the chat so you can find those. In addition, Holden's very own special education specialist, Sharon Graper, will be hosting a series of workshops on incorporating these materials into your classroom, starting on February 23rd, and we'll put a flyer in the chat for that. And we'll put Sharon's contact information as well. And other than that, I really want to remind you to not miss. I don't know how I'm going to get over each and every talk being just as fun as the next one, but Dr. Baranda Montgomery is going to talk about from seeds of inspiration to a harvest of discovery. And so that link to register for the next talk should be up on the Holden Forest and Gardens page. So check back with us again, and thank you everyone for coming.
Thank you so much.
Thank you.